Hi everyone, I'm very happy to have the opportunity to present our poster, Building Generalized Protein-Protein Interaction Models for Robust Out-of-Distribution Cross-Species Interactions Using RAPPPID. I'm Joseph Szymborski, a PhD student at McGill University's Electrical and Computer Engineering Department and a member of MELA, the Quebec AI Institute. I performed this research with Amin Ahmad, my supervisor and professor of electrical engineering at McGill University. Professor Ahmad is affiliated with the Goodman Cancer Research Institute and MELA, the Quebec AI Institute. Let's begin with a brief introduction. Our work here is motivated by the knowledge gap between model organisms and the much larger group of non-model organisms. Specifically, we're interested in the disparity between the size of their known protein-protein interaction, or PPI networks. We'd like to close that gap using a deep learning PPI prediction model. There's a challenge with making PPI predictions on non-model organisms. While they are the most likely to benefit from prediction models, they offer too little data to make predictions on. That's why it's desirable to train on model organisms with much data and infer on species with fewer data. Many ML models do not make accurate out-of-distribution predictions, which is something required to train and infer on different species. Let's move on to the methodology. First, to create a model which is able to make generalized out-of-distribution predictions, we must ensure our methods of validation are robust. Data leakage in PPI prediction tasks is a pervasive problem. It's common for PPI prediction methods to make their predictions on datasets created by random splitting. This involves randomly assigning interactions to training or testing splits. This has been shown to result in data leakage. Instead, all analyses we conducted are on a dataset where proteins were split into training or testing groups. Only interactions between proteins of the same group are permitted, ensuring no proteins in training interactions are found in testing interactions. I'd like now to introduce RAPPPID, a deep learning model for PPI prediction. RAPPPID is a regularized twin neural network that adopts a modified AWD LSTM. RAPPPID considers pairs of amino acid sequences with an interaction label, making RAPPPID a sequence-based method. Amino acid sequences are first tokenized with a sentence piece tokenizer. We then compute fixed-length vectors that represent each sequence using a bidirectional AWD LSTM. Latent vectors are then concatenated and are inputted into a two-layer, fully connected classification head. The output of the classifier is the interaction probability. To learn more about the RAPPPID model, please see our paper in bioinformatics. A citation is available at the end of this presentation. I'm now excited to present our results. We find that RAPPPID performs better than leading PPI prediction methods when making predictions on a dataset of human interactions. This dataset was prepared using the protein splitting approach outlined in the methodology section. Here we see that RAPPPID obtains higher scores than leading PPI prediction methods, obtaining an average AUROC of 0.767, average precision of 0.779, and a Matthews correlation coefficient of 0.371. These averages were obtained from evaluating these methods on 3 random training and testing splits. We also find that RAPPPID maintains high intra-species performance on various organisms. We trained and tested RAPPPID on 8 distinct organisms, ranging from mouse to Drosophila to baker's yeast. Here we find that RAPPPID achieves AUROC and AP scores that are largely comparable across the board. A result we were quite pleased to see was RAPPPID's cross-species performance. This graph illustrates that a RAPPPID model, which is trained on a human PPI dataset, maintains its performance when tested on other evolutionarily distinct species. On the y-axis, we have the Matthews correlation coefficient, and on the x-axis, we have the evolutionary or patristic distance relative to Homo sapiens. The unfilled markers here represent the performance RAPPPID achieves when trained and tested on the species indicated by the marker color. The filled markers represent the performance of RAPPPID when trained on human data and tested on the species indicated by the marker color. The dashed gray line indicates the performance of the human model when tested on human data. As you can see, the cross-species performance appears strong across nearly every organism, independent of evolutionary distance. We were also happy to learn that RAPPPID learns biologically relevant features. We tested whether RAPPPID was actually relying on the sequence of the proteins in the PPI graph, or rather taking advantage of characteristics of the PPI graph, like degree distribution. To do this, we randomly assign proteins the amino acid sequence of different random proteins. This maintains the degree distribution, but the network no longer makes any biological sense. A model that relies on the biology encoded by the protein sequence would not be able to perform well on such a dataset. RAPPPID's performance, as measured by AUROC, drops by 43% down to random chance when trained and tested on the scrambled dataset, indicating that RAPPPID relies on biologically relevant features. I'd like to take this time to acknowledge our generous supporters. Thank you very much for listening, and have a great day.